some slides that just give some updates on behalf of the Knowledge Portal Network, and then we'll hand it over to Trang, our featured speaker. So first, I just want to invite everyone to join us at the American Diabetes Association for their scientific sessions June 5th through 8th. We are very excited to be here another year. We are going to be part of the NIDDK Scientific Corner, booth 2324. We are very excited, as we said, this session. We'll be showcasing all of our portals, introducing some new ones, and we're eager to talk to you all there. So, as a reminder, the NIDDK Scientific Corner is going to feature various resources. These are all the resources that will be in person. We also have a scientific corner that will be online to visit some other resources. If you are interested in exploring that as well, we will share that to all of you when the time comes. Another event that we are very excited to be a part of this year is the Festival of Genomics, Biodata and AI. So this will be presenting on behalf of the CFDE. And so we will be there June 3rd and 4th in Boston. And we will be at booth 1A. You can learn more about our Knowledge Center that we're also funded through another mechanism. You can ask us any questions. We'll also be showcasing other members of the Common Fund data ecosystem. So please feel free to stop by and talk with us at this session. We also have a talk in room 50 on June 3rd in the afternoon, which is from Prompt to Provenance, an AI-enhanced contextual workflow across the Common Fund data ecosystem. And this might be of interest to you if you are interested in learning about how AI can guide researchers from natural language questions to structure knowledge graph-driven discovery in a way that can bridge data sets, ontologies, APIs, and literature. And so we're going to have various different speakers be involved in this session. So if you'll be attending Festival of Genomics, please feel free to stop by. Like I said, either the booth or our talk. We'll happy to answer any questions or give you any demonstrations on our behalf as well. One other thing that I just wanted to make you all aware of, there's also this funding opportunity called Pilot Projects Enhancing Utility of Usage and Common Fund Datasets. The reason we're showcasing this here is because I think there are some people that can also, see this as part of their research. This is closing June 23rd, and the idea is that there are many valuable and widely available data sets that have been generated by Common Fund programs. And so if you're interested, you can apply to demonstrate and enhance the utility of these data sets. Please feel free to scan the QR code here for any additional information. And one other webinar that I would like to highlight is through our Common Fund data ecosystem. On May 29th, you can hear from Illuminating the Druggable Genome, IDG, and the National Center for Advancing Translational Science, NCATS. They are going to be showcasing their Pharos 2 release and also go through some program updates as well. So this is another webinar to keep an eye out. And one exciting thing that we are happy to be introducing coming soon to our portal is the Knowledge Portal Network blog. And so this will be releasing most likely Monday next week. And this blog is designed to share scientific research, emerging insights, and perspectives across all of our various disciplines. Our goal is to make these complex ideas more accessible, meaningful, and relevant to the research community and beyond. So if you're early in your career, a seasoned researcher, or you're just curious, this will essentially have some insights from our Knowledge Portal team and some collaborators as well. We'll talk new discoveries, evolving trends, and just commentary from the voices in our community. So we're very excited about that. And our first blog will actually be from our featured speaker, Trang, which will be really exciting. So please keep an eye out. We're very excited to get this up and running. So with that, these are all the updates that I have on behalf of the Knowledge Portal. and we'll be able to have our featured speaker present. So, Trang, feel free to take it away. Thank you for joining us.
Thank you, Mackenzie. All right, I'm going to share my screen. Um, can everyone see my screen?
Yes,
All
thank you.
right, perfect. Hi, everyone. My name is Trang. I'm a computational biologist at the Knowledge Portal Network. Um, I've been working with everyone on the team for the past 5 years to ingest a lot of different types of data into the portals and work with the engineers and scientists to help visualize and make sense of these data. And B, I've also been working very intensely with rare variant association analysis. So that's really my expertise in terms of the technical side. So because I've only ingested the different types of data into the portal, the bad news is I don't really have a very deep understanding of the data that we're trying to explain today. But the good news is because I have surface level understanding, I will not go into too much technical detail. So, so if you have any questions or you want to help me understand and explain a bit more about these concepts that we're discussing today, feel free to just chime in and we're happy to discuss. All right. So I'm going to assume that most people here are somewhat familiar with GWAS, which is really the bread and butter of a lot of our knowledge portals. Um, so for the past 20 years or so, we have been able to sequence large-scale genotype across populations and associate different regions of the genome with different diseases and complex traits. And because of that, we've been able to identify a lot of GWAS loci that are significantly associated with disease. Um, a lot of signals are coming out because of the big sample size that we have. So, for example, this is the local zoom plot of one region around KCNJ11 gene associated with type of diabetes. Um, and the reason why this is still not enough for us to really understand about the biology or the mechanisms in complex traits is because you can see that a lot of variants in these loci are associated with type of diabetes, and it is extremely hard to pinpoint exactly the causal variants. All of these variants are in LD or linkage disequilibrium with each other, and maybe only a few of them are causal and the other ones are just like tagging along and we don't really know for sure. And the second challenge in translating GWAS loci to actual biological insights is because, as you can see, in this region, there's a lot of different genes. So, because type of diabetes has been really heavily researched, and we kind of know that KCNJ11 and ABCC8 are the effector genes for this disease, but for a lot of other disease and complexes, we don't really know for sure. And amongst all of these genes, we don't really know which ones are actually causing or relevant to the trait. And then there's a complication of linking the variants to the genes as well, because in GWAS, most of the variants are non-coding, so they are regulating genes around the area, but we don't really know for sure which genes they're regulating. Um, and to add more complication, usually diseases and traits are very complex, involve a lot of mechanisms, usually specific to cell types and tissues. So, for example, type of diabetes is in beta cells in pancreas and sometimes implicated in adipose tissue or liver. So not all tissues and cell types are equally relevant to a disease, and that affect how we interpret the GWAS signal as well. So today, because we're presenting the Variant Sifter to you all, I want to focus on the first challenge of the problem, which is to really pinpoint or at least prioritize the likely causal variants within a GWAS loci. So before I go into the Variant Sifter, which is the tool that we've developed to help everyone prioritize the variants, I want to kind of briefly go over the different approaches that the field has taken to help prioritize variants. Not all of these approaches are being implemented in the Variant Sifter, but we're going to go over them and discuss how we can integrate the rest in the future. So the first approach is called variant fine mapping, which is to um, just basically we try to get a better resolution into which exactly is the variant in this locus that is driving the GWAS signal that we see, um, or which variant is actually the causal one in all of these variants. The second one is functional annotations, where we ask the questions of, okay, so let's say we have a couple of variants of interest that we are more confident than the others that are driving the signal. Do they actually fall into any meaningful region of the genome or not? The third approach that people have used is to, again, kind of ask whether these variants actually regulate any of the genes in the same in the locus. The fourth approach is quantitative trait loci or QTLs, which conceptually are very similar to GWAS in a way that we are also trying to conduct association studies between the variants and the phenotype. But this time, instead of the clinical phenotypes, we are doing the association between the variants and the molecular phenotypes. And I'm going to go a bit, into the details of these in the later slides. And then the fifth approach that I want to talk about today, which is very, really an extension of QTLs, is co-localization that asks the questions of whether the variant in question or the variant of interest is the one that's actually driving the signals in both GWAS and QTLs. So ideally, in a association studies, when you identify a significant loci and you're trying to figure out which variants are causing the signal, um, we want to combine different lines of independent evidence together to support that prioritization of that variant. So the first approach I want to talk about today is variant fine mapping. As I mentioned before, the idea is pretty simple. Conceptually, it's just asking the questions of among all of these variants that are significantly associated with this trait, which ones are more likely than others to be causal. So in this method or this approach, we need GWAS summary statistics. We need LD reference panels or in-sample LD, which just tells you which variants are correlated with each other and hopefully identify um, the, the main or the lead variants and distinguish the lead variants from the other variants that are just tagging along. And some common methods are FineMap, SUSY, Cavia, or Paintor. So this is just a very simple conceptual model of how this works. If you look at the figure on the left, the upper panel is just a like a scatter plot to show the p-value between a variant and the trait. And then the figure below is the PIP, which stands for probability posterior inclusion probability of the variant. So basically, from the GWAS locus, we kind of try to identify the different small sets of variants that are likely to be independent signals from each other because the region is pretty fuzzy. So we try to find different chunks of variants that are not really correlated with each other and show independent signals. And then within each of those chunks of variants, we kind of identify which variant is the most likely to be causal. So the PIP is just the probability that tells you that. I see some comments in the
Yeah,
chat,
I was
Mackenzie.
just gonna say, Trang, there's a question.
Yeah.
So in slide 3, you had mentioned 5 layers of evidence for prioritizing variants. Usually, how many layers are considered reliable enough from your experience?
Yeah, so ideally, it's actually a question. The Variant Sifter is exactly for that. Ideally, we want as many po as possible. Um, a lot of these are not really straightforward to conduct. So for example, like for QTLs, you need to kind of have for example, gene expression across different cell types and tissues. So a lot of these involves actual experiments you have to conduct, which can be very expensive. So we have a lot of like databases, the public databases that have provide these variants, these evidence already. But in a study, ideally, people probably would want to conduct their own experiments to get more, I guess, fine-grained evidence. Um, so yeah, I would say it depends on the study, but obviously the more the better. And I think what matters here is that these are they should be independent lines of evidence. So you don't want, functional annotations, for example, to be the same source of data as the links to regulatory elements.
Great, thank you.
Yeah. Um, so then, sorry, we stop at this. Okay. So yeah, so in variant file mapping, the output is just the credible set, which is a list of variants that are in LD with each other. And then for each of those variants, you have one probability that indicates how likely it is to be the causal variant. Um, so that's variant file mapping. The second approach is to evaluate, sorry, the second approach is to annotate the variants of interest with functional evidence. And functional here means a lot of different things, but in this particular context, the first meaning of function is coming from the bioinformatic predictions of the variants. So usually we have VEP, which is a very popular tool to annotate the functions of each individual variant. So for example, we have conservation scores that tell you how evolutionarily conserved this variant is in mammals or vertebrae. If it is highly conserved, then it is more likely to be important. Actually, sorry, if it is highly conserved, then the disruption of it would be more likely to cause some phenotypic changes. Um, and then the next type of functional annotations that we have are not really variant based, but it's almost region based. So it talks about the functions of a region in a locus. So we have chromatin accessibility, we have histone marks, which can be translated to promoters or enhancers, and we have transcription factor binding. And this is just like a brief graphic of how these can help us, like how these are involved in helping us prioritize the variants. I'm going to try to explain this very briefly. So all of these three types of data, chromatin accessibility, histone marks, and transcription binding, they are involved in gene expression. They regulate the expression of a gene, which then downstream affect some disease or trait. Um, so it's actually easier to go from the bottom up. So if you look at the very bottom panel, you have the promoters, which is the start of the transcription. So the promoters are where the gene starts to be transcribed to RNAs. And then transcription factors are the one that are, deciding the activity of the promoters. And these transcription factors have to bind to some region, some remote region of this gene. And so that's where the binding sites come in play. And then the binding sites are part of the enhancer region that are similar to like a remote control of the gene transcription. But in order for the transcription factors to bind to the enhancers or to bind to the binding sites, the actual chromatin of the whole DNA has to open in the first place because you know how DNA is like kind of twisted and like it's a very complicated hairball. And so that area where the transcription factors can bind have to be open in the first place. So that's what we call the open chromatin regions. So all of these kind of work together in a very complex manner to regulate gene expression or gene activity downstream. So you can imagine that if you have 10 variants that are significantly associated in this locus, and then only two of them are actually falling into um, any of these functional regions, then maybe those two you are more confident that they are actually doing something to the phenotype than the other eight. So really, functional annotations are a way for us to add more biological context to the variants that we're looking at. The third approach is very similar to functional annotations, actually. The third approach is kind of assess whether these variants are actually regulating the genes of interest or not, or the genes that we're thinking to be the relevant genes to the disease. So it's very similar to the functional annotation in a way that they're both asking the question of, is the variant actually functional or functionally meaningful or not? But the third approach directly links all of these regulatory elements that we just talked about in the second approach to the actual genes in the same locus. Um, so the first, there's many different methods to do this. The first one is the simplest one, which is just, figure out which the gene is the nearest to the variant. So you just kind of get the position of the variant, get the position of the gene, and then see which gene is the closest. And oftentimes it is correct, but also, also often it is incorrect. So, for example, this is a very classic instance where the nearest gene is not always the gene that the variant is regulating. So in this picture right here, from a study, from a GWAS study of obesity published almost more than 10 years ago, um, the researchers found a lot of different variants that are associated with BMI. And even though they are in the intron region of FTO, they're actually regulating a gene that is to the right of FTO, which is IRX3. And then later on, over the past 10 years, we've also had a lot of different studies that confirm this initial hypothesis that these variants in these locals are actually regulating IRX3 and not FTO. So this is a prime example of how the nearest gene is not always the correct gene that the variant is affecting. But it's often the simplest, so we still use it as kind of one method to prioritize variants. The second approach is kind of figuring out using a lot of different experiments like ABC model or Hi-C contact maps to figure out if like spatially this variant is actually falling into a region that is regulating some genes in the locus. And then the third line of model, which is probably the most um, which is the line of evidence we're most confident in if we have access to the data, is perturbation. So you would directly just change, like base editing a variant or perturb a region of the genome and just see how that affects a particular gene. So that is like probably the golden evidence that you can use to be most confident about. But obviously, this is very expensive to conduct. So that's why we don't really have the capacity to do this for every single variant across the entire genome. Um, so then the summary of this approach is just, we're trying to figure out which genes that these variants are regulate. And because of that, we can also use this to also identify likely effector genes as well. The fourth approach is quantitative trait loci, or QTLs for short. The idea is very similar to GWAS, where you also try to test the associations between the variants and the phenotypes, but this time instead of clinical phenotypes like disease or complex traits, you have molecular phenotypes. For example, you have gene expression, you have splicing event, you have chromatin accessibility, or protein levels. So then, each of these is their own phenotype, and you conduct again like association studies between the variants. So for example, this is a violin plots from GTEx for IRX3. And the violins plots are just showing the different values of the y-axis is the different expression values of this gene. And then the x-axis is the different, values of the genotype. So it's whether it's CC, the referent is C, right? So whether both alleles are C, or one of the alleles is C and the other one's T, or you have both alleles being mutated from C to T. So then you just kind of conduct maybe regression analysis to figure out whether the genotype, the changes in the genotype of a variant is associated with the expression values of this gene or not. And this is often tissue specific. Um, and because of that, not only this approach can help you prioritize variants, but also and identify likely effector genes, but you can also kind of somewhat identify the relevant tissues as well. So especially if you, so it's almost like all of these approaches are supporting each other, right? So if you have a variant that you're confident in, and then you see that, oh, this variant is also associated with the change in the expression of a certain gene in a certain tissue, then that gene and tissue is also something that you should be interested in studying further. And then the final method that I want to talk about today is co-localization. So this is really the combination of GWAS and QTL that I just talked about. Um, it's basically just asking the questions of, okay, so we see, associations in GWAS, and then you see associations in QTLs, and you want to compare that with the same locus. Are these signals the same with each other? So it's like asking whether the same variant is driving the significant signals that you see in both GWAS and QTL. It was a very kind of generic graphic again of how this works together. This is from this is a figure 2 from eCavia, which is the one of the newer method for colocalization, and it's showing the different scenarios between GWAS and QTL signals. Sorry, before I go to so in each of these panels, the triangle is showing the LD scores of the variants with each other, and the darker the square, the higher the correlation. So in figure A and B and D, you see that the squares are not very dark. So it just means that in this region, the variants are not really correlated with each other compared to figure C, where all the squares are very dark. And then in the actual scatter plots themselves, in each of the panels, the upper sub-panel is, I believe, GWAS, and then the panel below that is the QTL association p-values. And then the x-axis is the different SNPs or different variants in that particular locus. Um, so then in scenario number one, you can see that the association patterns between GWAS and QTL are very similar to each other, right? You can see that, if a variant is super significant in GWAS, it is also pretty significant in QTL. And all of these variants together follow pretty similar patterns. And then because in this region, the variants are not in too much LD with each other, so a lot of these variants are pretty independent from each other, then you can sort of be confident that the variant right here, like the top variant that has y-axis, y value of 12 and 14, is probably likely the causal gene that is driving the signals in both studies. So then you have the CLPP, which is co-localization posterior probability being very high. And then, if you see high CLPP, then you're again more confident that this variant is what's causing the signal in both GWAS and QTL. And then panel B is when in the same locus, you have different variants causing the signals. So the association patterns between the GWAS and the QTL is no longer the same, right? Like you can see that um, on the left, like at the beginning, like these variants are similar in patterns, but then as you move down to the right in the tail of the plot, um, a lot of variants are significant in QTL but not significant in GWAS. Um, so it indicates that both GWAS and QTLs they don't really share the same causal variance in this region. And then panel C is when it's kind of a, a different scenario from A and B, as in that, it is very similar in A, because again, like both studies follow pretty similar patterns, but then because you have a lot of variants in LD with each other, like all of these variants are super correlated with each other, even if you know which variant even if you see that a variant is the most significant in both GWAS and QTL, you can't really tell if that's the causal variant or not, because again, it's in LD with other variants that are also significant in the same locus. So then your confidence is much lower for this variant. And then panel D is a situation where you have multiple causal variants in the same locus, but because the variants are in low LD with each other and the association patterns are also similar, eCavia can actually also kind of detect that um, there are two causal variants in this region with high confidence. So then this method is really trying to kind of distinguish the shared causality from coincidental LD overlap, right? So we can kind of tell you how confident we are that the same variant is um, associated with a disease from GWAS and also associated with, let's say, the expression of a certain gene in a certain cell type. Any questions so far before I move on to the next part of the talk?
Alan, go ahead. I will take you off of mute.
So I have a question. I often, when I'm looking at the T2D portal, there'll be one single SNP that has a very, very strong p-value, uh,
Yeah.
and no others around it. And my instinct is to ignore it. Um, but in light of what you just said, you're really suggesting that if there aren't other SNPs in LD or whatever, that maybe that's a significant one, and yet I still feel like it, an isolated SNP should be viewed with a lot of skepticism. What are your thoughts about that?
Yes, I agree with you actually. Because the LD is both a blessing and a curse, right? It's the same thing in exome studies as well. If you're seeing like a super strong exome like coding variant signal, but then around it there's no non-coding variants, then it's something that you should look into deeper to make sure that it's not just noise or like artifact. So I do agree with you that we should, be careful when we interpret or trying to like definitely say that that variant is the causal variant in that region. But at the same time, um, we have like the sample size for GWAS for type 2 diabetes has increased so much that mm, when a variant is like super, like let's say like 50 orders of magnitude more significant than the other variants. But then you see the other variants in the same region are still somewhat like genome-wide significant. Um, I would have a hard time believing that that is just noise. Right?
Well, are you familiar with any examples? I'd be curious to know if you've ever seen an instance where there's a single SNP with a p-value of 10 to the minus 50
Hmm.
surrounded by SNPs with 10 to the minus 9 or something, and that SNP actually does end up being proven to be a causal SNP. Have you ever seen that?
Um, I have not personally, but maybe my other colleagues in the call can speak to that.
Sorry, I didn't mean to
Anyone?
put you on the
No,
spot.
no, no worries. I know this is actually I kind of now that you mention it, I want to. Do you have a specific like example for a gene?
I see them quite often, but I can't come up with it right this instant. I
Right.
I
So
do see
one
that. And it,
one
the other problem, and I'll just if I can just say one more
yeah,
thing.
yeah.
One of the problems with the T2D portal, I feel, is that when it lists SNPs beneath the regional plot, it lists them by LOD score without regard to proximity. And so sometimes it'll show you 10 to the minus 50, but it's 10 megabases away. And so that's there should be a filter for that. That's just getting in the way of the whole analysis anyway.
I see. Yeah, that's a good point. So I'm gonna address two of those aspects.
Yeah.
The first one is that um yes, thank you. Someone put in the chat that the SNP is in the coding region. Yes, that is very
They
possible.
usually are not coding SNPs, though, I
Oh, I
have to
see.
say.
Yeah,
If it were a coding SNP, I would definitely focus and have, really focus in on that. But they're almost always not coding,
right.
actually.
So one thing I would suggest that you do in that case is to look into because on our portals we meta analyze a lot of different published GWAS. So if you see that odd signal that's coming up like too high, right, compared to the other variants, you should probably look into the individual studies that go into it and see if there's any one specific study that is like completely just driving the signal. And then if you see one smaller study that's like have 10 to the minus 40, for example, and the other ones are not even nominally significant, then that might be an artifact from that one study. But if you see like, you know, replicable signals across different studies that go into the bottom line analysis, then I would probably be a little bit more confident in that signal. Does it make sense?
Yes, thank you.
Yeah. And then I think the second point that you made is a really good point about um, the order of variants, right?
Yeah.
So on the portal, we have a lot of different pages and tools to help you kind of, identifying the genes almost. But they are not, obviously it's different researchers ask different questions. So it's kind of hard to make sure that all the information you need is one single place. But I would definitely check out the other genes and pages to get more information about the genes around the variants that you're interested in.
Well, it would be, I think, simple to insert a box where somebody could type in the coordinates, the limits.
Yeah. Yeah. Thank you. Yeah, thank you for the question and comments.
Thank you.
Yep. Anyone else who would like to? All right, I will move on then, and
Yeah, I
then
was gonna say, Trang, I think you can go on and then
yeah,
I'll stop you
perfect.
if anything
Thank
else
you.
comes up.
Yeah. Anyway, so I think the second part of the webinar today is I want to kind of demonstrate how you can combine these different types of evidence to prioritize the variance in the Variant Sifter, which is a tool that we've developed. It is still in beta mode right now, so we're hoping to um, develop it further. And at the end of the webinar, I also would like to take suggestions from the community to see what would benefit you the most in this analysis. So uh, before I go into the tool, I want to clarify that right now we only have the first three sources of evidence. So we have variant fine mapping, we have functional annotations, and we have linkages to genes and regions. We don't have yet QTLs and co-localization in the main variant sifter, although we do have some of that like elsewhere in other tools, but not in the variant sifter yet. Um, Actually, I'm gonna try to do this live. That would make it maybe smoother. I preloaded the website, so it should not crash.
Can also send the link as well for people to explore in the chat too if they're interested.
Yeah. How do I do this? So let me go back to my Chrome tab.
While we're waiting, any other questions?
All right, here it is. Um, so I preloaded the page already, but I will kind of walk you through how to get to where. It's it's all in one page, so you don't have to like, really go around at all. The first thing that you want to have is just the phenotype of interest and a gene or region that is associated, that has some GWAS association with this phenotype, because they're trying to like identify the causal variant in this region, right? Then, sorry, one second. My Zoom is popping up a bunch of different things and I can't see the screen. Okay. So when you enter a gene um, or region of interest, it will first show you the local zoom plot. So this is just the GWAS association statistics within this region. And then you can also filter by variant ID or p-value, or beta or z-scores, or the consequence of the variant as well, to kind of uh, make the plot a little bit easier to digest, if you have something in particular that you're looking for. Um, and then obviously this is the raw signal from GWAS, right? And again, this is coming from the bottom line analysis from the portal that we generated by meta-analyzing all the published GWAS for a trait. Um, and as you scroll down, as I mentioned previously, we do have fine mapping results on the variant sifter. As you scroll down, the the genome, what do you call this? The genome coordinates stay the same to help you kind of navigate the variants of interest a little bit more easily. So as you scroll down, you can select the different credible sets from different fine mapping studies. For example, here I'm choosing these results from the Mahajan study in 2022, which I believe is one of the largest GWAS studies so far. And you can see the file mapping results from their study. So the variant that I have right here, um, actually that I start right here, is showing up down here as a star as well. And when you hover on it, you can see the exact values or the exact results of the file mapping. So this variant compared to the other variants in this credible set has a much higher posterior probability. So basically, it's just telling you that it is likely to be the causal variant in this particular region, according to these results from a particular study. Um, and then as you scroll down, you are going to see the functional annotations, which is the third approach that I just talked about. But because the functional annotations are often tissue specific, we first show you the global enrichment. This is results from um, the SLDSC method that we implemented on the portal. And it's kind of telling you that, for each tissues that we have available, which functional regions are enriched with GWAS signals compared to the baseline. So as you can see here, pancreas, not surprisingly, because this, with type 2 diabetes, you have pancreas, you have liver, that is coming up as the more significantly enriched tissues across the different annotations that we have. Then that kind of help you narrow down the focus a little bit for the tissues in which you are going to look into the functional annotations. And then as you scroll down, because I kind of marked, like I marked the variant at the beginning of the local zoom plot, it's going to carry like all the way down over here. So the vertical red line here is the exact position of that variant. So at the quick glance, you can very easily see whether this variant is falling into the accessible chromatin region um, in the pancreas tissue or not. Um, and it looks like it's like borderline, it's like there, but not really. Um, so this is kind of one quick way for you to figure out how a line of evidence to increase or decrease confidence in this particular variant. And then if you click on the track of the pancreas, it's going to break the tissue down into the different cell types or biosamples that we have. And all of this data is coming from our sister resource, the Common Metabolic CMDGA, the Common Metabolic Disease Genome Atlas, where they integrate and harmonize across all publicly available functional data. Um, so then, similarly, you can also just click on the different annotations and see whether or not this variant falls into that region of a tissue or not. So if you click on binding sites, for example, um, the binding site tracks are going to show up. And then as you scroll down, I think the last piece of data here is whether or not the variant is linked to a gene in the region, or whether or not the variant is falling into a regulating region of a particular gene. So you can see here the first tracks in yellow right here is the SNP to gene data that we got from, I think, a paper published by Arkis Price's group, where they combine different experimental data to infer whether or not a variant is linked to a gene. Um, so similarly, to the other tracks above, you can also see the vertical line here, which indicates where the variant is in the genome, and like just visually, glance whether or not it's falling into whether it's linked to a gene or it's falling into the regulating region of the gene in a particular tissue. So for example, in here, it seems to, I should probably click a different variant to make sure to see it would be better, maybe. Uh, let's see this one. Yeah. So for example, so maybe the second variant that I star is you're probably more confident that this variant is doing something than the first one, because the first one's really not really having any evidence at all, whereas this one is falling into all the tracks and having all the evidence line up pretty nicely. Um, so yeah, that's how you can kind of combine different types of evidence to prioritize variants in a locus. And then the final section of this tool is a table that summarizes all of the information above, and you can also see the exact data sets that are contributing the results. And when you click on them, it's gonna lead you to the exact data set hosted on the CMDGA, and then you can find out more about that particular data set. Um, Yep. So that is kind of the current development of the Variant Sifter. Uh, in the future, we are planning to also add the other types of data as well. For example, like we are going to add the bottom line ataxic or QTLs or co-localization. And we're also going to add some new methods. For example, Fagan is, I believe, a method that we're developing to kind of find the genes that are driving the signal in the locus by linking the variants to a gene like more confidently using different data types. And we are also thinking of integrate AI into this tool because it is a quite complicated tool with lots of different data types, and not everyone is familiar with all of them. So having a reliable AI agent that can help you sift through all of these data would be ideal. But we just have to make sure that it's not producing any unreliable results. Um, Before I end the presentation, I would like to highlight that the Variant Sifter, even though it's still in beta mode, we are already getting a lot of interest from other collaborators in using the Variant Sifter or some variations of the Variant Sifter to support their research. So one example is the Giant Consortium. Um, the Giant Consortium has been doing a lot of the genetic studies of anthropometric traits such as height, BMI, waist hip ratio, and they're currently using, we've developed a specific portal for them with the Variant Sifter to kind of browse their own GWAS as well as their own functional data. Um, And the second collaborators that we've been working with is Ayla Segre, who is an expert in um, vision genomics. So so we developed this portal for her to also use a lot of the tools and functionalities in the Variant Sifter to visualize and integrate her QTL data, co-localization results, and others data type. So all of these collaborations really kind of have helped us understand where the needs are coming from. So hopefully we can also get your suggestions and ideas to develop this tool further. And I'd like to thank everyone on the team who has been working really hard on the tool and making it both accessible and very, visually stunning and easy to navigate. So if you have any questions, I'm here to discuss. Thank you.
Thank you, Chong, for that presentation. If you have any questions that you would like to ask Chong or the team, please feel free to use the chat, question and answer, or raise your hand, and I will give you the option to come off of mute. So I'll just give it a second. Also, if you think of any other questions after the session, please feel free to email us and we can answer any questions directly as well.